Please again, you go from Sorry.
I have a question, I guess, about Italy and the UK. Is there any person who presents to our emergency department?
Dio santo, non è mica semplice. So sì.
Sir, can you?
Il mio. Scusate.
Yeah, so some folks with people with psychosis that are not actually talking, just have to remember people, but illustrating this point again that, one talking about how their visual and auditory hallucinations stop them from getting sleep, and the other about how it worsens their symptoms. I think Martha spoke to that, her clinical experience with that, that this is a real major area of interest for patients as well. Oh, sorry. And again, as we've covered, that sleep or sleep disturbance is not just one trait. It's multiple overlapping domains that together contribute to a sort of picture of sleep quality. And deficiencies in any of these areas is associated with a huge range of psychiatric and physiological outcomes. So we know that sleep is tightly woven to mental health, but we still don't fully understand how and why. And this is where I think genetic data is really useful. So both sleep traits and psychiatric traits are heritable, which means that genetic data gives us an opportunity to try and address some of these, or try and get this really understanding this relationship more. Our genes are set from birth. This is a major benefit that we have in the genetic literature. So you know the direction of the effect, or not from birth, actually, from conception. So we have that temporal understanding of these relationships, which we don't have in the observational literature always. We can use genetic data to get at the underlying biology, to understand more about the biological mechanism, and we'll give some examples of that later. And genetic data lets us move beyond just looking at single traits in these diagnostic boundaries, and can allow us to take a multivariate and potentially transdiagnostic approach to these complex phenotypes. So just to orient everyone with what we mean when we say something's a heritable trait, this just means that some of the proportion of the variance that we see across the population in a given trait is explained by genetics. And I made this quick figure trying to pull together a lot of the existing studies on the heritability estimates. You can see these bigger orange bars are estimates from twin studies, which we'll talk about briefly in a minute. And you can think of this as almost the sort of upper ceiling on how much of the population variation can be explained by genetics. And you can see quite substantial heritability estimates of for some traits up to getting close to 50%. If I was to show you this equivalent figure for psychiatric traits, those estimates are even higher. The yellow bars is our SNP-based heritability. So this is the amount of the variation in the population that can be explained by common genetic variants that we've discovered so far. And you'll notice a big gap between those two estimates, and this is expected or common. We see this across the literature. This kind of just means there's a lot more to be discovered in the last year, which is really exciting. And sleep is a complex trait, as almost all traits, but sleep and every way in which we measure sleep or sleep quality is a complex trait. So there's no single gene that makes you a bad sleeper. Each sleep phenotype is influenced by hundreds or even thousands of common genetic variants, which each have a small effect on their own, but taken together can have a really substantial impact. And of course, the environment matters as well. So complex traits are influenced both your genes and the environment. And we know that there are many, many ways in which different environmental exposures can impact your sleep. And the relative contribution of your genes versus the environment can change across individuals and across the lifespan. So just going to twin studies. So this is one of the most sort of the original genetic study really, pioneered by Francis Galton, who made other more questionable contributions to the literature, but twin studies is a sort of enduring contribution, and it's basically a natural experiment that takes the idea that if you have identical twins, monozygotic twins, we know that they share 100% of their genetics, versus maternal or dizygotic twins, which share on average 50% of their DNA, just like any siblings. But since both types of twin pairs will share the same environment, so, same household, same parents, you can assume that the amount of the shared environmental contribution is equivalent. So if you compare identical and non-identical twins, and we see that the identical twins are more similar than the non-identical twins, that the extent of that greater similarity it must be explained by genetics. So that's what we can build up these models to compare that, and there's varying different models you can build, different levels of complexity, but that's essentially what they get at, is comparing non-identical and identical twins and quantifying, passing out the shared environment, or the additive genetic effect, and then it's non-shared environment. So it's a huge literature, and there's a lot of work in our department. I'm based at King's at the SGP, which has hosts one of the largest twin studies, the TED study. So there's a lot of work in our department and across the rest of the university on twin studies. But I thought I'd highlight just a few here briefly before moving on to some molecular genetics, which is more my area. So firstly, the twin study showed before we had any molecular genetic data to prove the same, that a large proportion of the comorbidity between sleep and anxiety and depression can actually be explained by genetics. So some of the earliest evidence of a shared biology. Secondly, that genetic influence on sleep in childhood can predict psychiatric risk in adulthood, which I think was touched on in St. Foster's talk as well, that maybe sleep can be sort of show up earlier, and actually this persists even at the genetic level, which has implications potentially for early intervention strategies and that sort of thing. Thirdly, that sleep traits, there's difference in different sleep traits associate more or less with different psychiatric traits. It's not a sort of homogeneous association across all the different pairs that you could look at, and actually there's more sleep traits are more strongly associated with internalizing disorders than externalizing disorders, for example. Twin studies have also confirmed and seen that genes don't explain everything. So there's, even after accounting for these shared genetic factors, there's this residual environmental correlation between sleep and psychiatric phenotypes. So this tells us the relationship isn't purely genetic, it's not purely due to shared biology, which suggesting evidence of sleep and or psychiatry sitting along the causal pathway and these feedback loops that we heard about. Finally, to highlight that sex matters, heritability of insomnia, for example, is higher in women than in men, and the genetic correlation estimates can look different in sex analyses. That's something that's come up quite a few times at the conference so far, so that's also something that twin studies have contributed to. So twin studies will give you a really good sort of idea of where there's going to be some genetic relationships and genetic dating and probe, but they can't tell us the actual molecular mechanisms and biological pathways that are driving that, and for that's where we need to move on to molecular genetic approaches. So I always think it actually is easier to introduce genetics in some ways by talking about rare variants, even though That's not the area I work in. But most of us, when we think about the genetics of diseases, particularly, we're more attuned to thinking about rare variants, where we think maybe the Huntington's variant or something, where there's a single mutation in a gene that has a very large deleterious effect. And there are examples of this in sleep and psychiatry. These are mutations that tend to be present in less than, well, it's still defined as mutations present in less than 1% of the population, but very often we're learning things from variants that are seen in far less than that, maybe only even seen in one family that we know of, so they could exist and not indefinite. Often it might just be one family that sort of is studied because they have a specific syndrome type or specific clinical symptoms that then lead them to be investigated. So again, just highlighting a few examples where this came up. So some of these genes that we heard about earlier, the HER2 block gene, so a mutation in a phosphorylation site in this gene, which resulted in this familial advanced sleep disorder, basically. So it shortens the circadian phase by about 4 hours, I think, having this just single mutation, so a really large effect. The PRY1 gene, another clock gene, so a splice variant in egg had the opposite effect, so it gives you this delayed sleep phase and lengthens the circadian period. DEC2, DEC2, which is a transcription factor. This is yeah.
I was thinking that for the rare mutation, have they been identified because there were some genetic syndrome where, you know, child or person had particularly disturbed sleep and then it was traced back to some of the genes, or these are rare mutations that are only relevant for sleep disturbance, but they're not other correlations.
My understanding is, at least for the first two, that these are all just sort of described as familial traits. So I think they've arisen from studies of specific families, and I think often, I actually don't know if that's the case in these examples, but very often it is that you have like a syndrome type thing or a really unusual phenotype, like no pain sensitivity, I think that capsaicin gene, that's how some of those were discovered. But yeah, I guess the main thing to highlight about these is that because these do have such a large effect, then you can spot them in clinic or clinically. And that then, even though most people don't have those variants, by seeing where something's gone wrong and had a major effect, that obviously tells you quite a lot about the normal functioning of that gene. So this is DEC2 gene, for example. I think this discovery was one of the first things that led to sort of some of the understanding of the relationship between sleep duration and the orexin system. But yeah, so as we said, these are rare. They represent they offer large scope for learning, but they actually impact a very small number of people. So understanding the reason why still most of us we may or may not have some rare variants that affect our sleep, but most of what makes our sleep different is explained by common genetic variants, which we want to know. So genome-wide association studies, probably most people are familiar with them, but just in case they're not. This is basically an idea that you can look across the whole genome, roughly, or at least look across the whole genome, the parts of the genome that we know vary. So the whole of our genome is about 3.2 billion base pairs long. Most of that, like 99% of that, is the same between all humans, and indeed, a good proportion of that is the same between all living things. But we're obviously all really different in lots of ways. So even though it's only 1% of our genome that actually varies, that's the 1% that we study, we zoom in on. That's where we can have a sort of scope to understand a bit more about what's making us different. So this is what a GWAS is. You basically look in those there are areas of common variation, single nucleotide polymorphisms or SNPs. You can look across the whole genome, and you're effectively running a series of like a million regressions, where you're saying, if I have this variant or this variant at this specific position, how much does that impact my outcome? Let's say morningness preference, for example. How much does that predict whether or not I say I'm a morning person? I don't know. We run these multiple times and you end up with a plot that looks sort of like this, where each dot represents a single nucleotide polymorphism, a SNP, and they're organized on the x-axis in terms of their physical position on the genome, and then on the y-axis the minus one p-value, so that those with the strongest association rise to the top. Robin had an example of this as well, so the strongest associations stand out like peaks of like the Manhattan skyline. Each of these variants on their own has a very small effect size, but if you look across all of them, then can often give you a real picture of the biological pathways, the genes and pathways that are involved in a specific trait. We need really large samples for this, and you sort of see this throughout some of the examples where you ideally really will be into the tens and into the millions, maybe even more, to really close some of that missing heritability gap. So there's always a trade-off in US studies with the need for this huge scale and the sort of depth or quality of the phenotyping. So here is some Manhattan plots from some of my work on sleep duration. So here is looking at sleep duration as a continuous trait, and even this variant with the largest effect size, when you actually sort of work out how much difference they're having, then each individual one is maybe altering your sleep on average by 1 or 2 minutes. I think 2 minutes was about the largest effect size we saw. But in theory, if you had all of the sort of negative or sleep reducing variants, that could have an impact of up to over 2 hours on your sleep and vice versa. Yeah.
I think this is a very good question. But thinking of the also topic showing the videos of terminal.
Yeah.
What the same genes are conserved across different species and plant. I think we can know what is different about genes and it's like the sleep.
I assume there is something known about that. In humans, the diurnal preference or the sort of sleep timing genes are quite biologically distinct from things like sleep duration. That's what we've shown in later slides. I don't know if you know much about what we know about animals.
Well, I think the fascinating thing is that in mice, the molecular pathway is the same. So the see what change of diurnal gas in nothing alone. And what that's used was so that's why in fact studying the acidic gas is really quite useful because it replicates the inhibition. It's downstream.
And going back to Robin's, sorry, I'm sitting here because I'm resting prepared to the wall. Sorry about that. All time. I'm not sleeping, I'm awake. How much epigenetic might have a role in switching on and off some of those things, even also muscular cross species. The environment and muscle leads and the sort of evolutionary habits that allow among the tribe. How much has that impact on some genes that should keep them away? Allow them to go to sleep. I'm thinking about the one that I donate, or I don't know how much epigenetic has a role in it.
Well, it's true. The stream is in the
Wow.
otherwise from evolution. So is that in this again. They need to have the ability which actually I think so another thing to violate is that then and more than that the government has not Well,
I have a teammates I have lost.
It's elevated, but you think we don't now and I work in for and the other machine is pretty much the same. Genetically, we can put this, then this, and some of them even actually passive and the center for the family, you can one that is just related to the environment. So just to which is absolutely, I mean, in built to the point that students and the and some bacteria in said different genes, but actually they're interesting. And so it's a whatever. As this, shock is already and he separated that. The vision was not the shit. So you soft and they are and if I want to change people to stand, they also they like sending about me. Which is the idea, is that it's an extraordinary issue. So
I haven't even touched on animals like this, so I don't know, so,
But
It's
it's
so-
so months.
yeah, true, but it is so interesting, and, amazing that something can be around for so long, and there's so much that we don't know.
well, it's another price of cancer change thing, and they're talking medical. But
Yes.
in rats, I know you made a people that mistake. It was decided.
It was this-
Well, no, that.
the same, yeah.
So, anyway,
That's-
It's quite interesting when I think about the philosophy, I don't think about sleeping philosophy like that. I guess, is there any animal that doesn't sleep?
so my people say is, and they'll stereotype my sleep habits, is that it's a period of, a period of physical innovation. Just something living around, living environment, which are, that, during the cat and develop a possible, and I think that definition works, that's already defined in decent, so this argument is accumulated, and you will actually be part of stimuli, to, use the, and push it forward about using the tree. It's something that, so it's just scientists put it out in time that it gives us a 50-minute lecture on secret languages. And it's wonderful, it's all about what they're doing with their
What's the lifespan? What's the lifespan of your cellular?
So in that condition, they can be up to 3 months.
Oh,
Yeah.
5 weeks is a lot.
yes, no, I didn't want to like a couple of ways.
The circadian is very similar to human.
Yeah, wow.
Therefore, don't have any, well, 1 p.m.
Did I?
Or 1 p.m. Then it's this, even the length, the period.
Still
It's 3.5. So it's probably just been considered for children that it's a picture that you just something that there's a base, and that you see that the big changes in scaling up just 2 or 3 days before the simulation when that comes up. Oh, it's a bit of a mean. So the period changes the magnitude. So it's about
No, it's so fascinating. Well, yeah, so sleep duration, so, and then, yeah, this is sort of continuous sleep duration, and then here we looked at long sleep versus, on the bottom and short sleep on the top, so actually quite different genetic signals when you look at it. Well, some similar genetic signals, but for the downstream analysis, this sort of ends up mattering quite a lot. So just to pull out some of the top hits, the top and most well replicated hit is in the PAX8 gene, which is a transcription factor that controls thyroid function. I don't know that there's much agreement on exactly the mechanism by which that would be associated. I mean, yeah, I don't know what the current thinking is on the role of the thyroid in sleep, that, sort of some sort of hormonal regulation or something, but this does seem to be a functional variant in terms of how all the analysis that's been done on this. FOXP2 is sort of even less obvious why that would be sleep in a way, but this does come up in a lot of our analyses of psychiatric traits. We also see the DRD2 receptor, which obviously is again very relevant to psychiatric traits. And when you look at sleep as a continuous trait, I don't think this will actually be a surprise to either sleep scientists or clinical psychologists, clinical colleagues, that there's a real non-linear relationship. So if you're looking at sleep, if you quantify sleep duration just as the number of hours you're sleeping in a day, you're masking quite a lot of heterogeneity there. But then, yeah, looking at lots of psychiatric traits, you can see lots of examples where we have high genetic correlation between both long and short sleep. So I think this is generally often what people see clinically, that there's not just one sort of altered sleep in psychiatric conditions, doesn't just look one way. So the chronotype was touched on earlier, and this is definitely in terms of the GWAS data, this is where the circadian genes show up. Yeah, that's the most recent GWAS. I think there's probably going to be a newer one coming out soon, but there was 16 genome wide significant loci, which match genes involved in photoreception and circadian rhythms. And as we heard about earlier, the relationship between these clock genes and psychiatric diseases is also well established. A lot of these also separately come up in the GWAS on depressive symptoms, anxiety symptoms, for example. As just to touch on the phenotyping, obviously, when I say chronotype here, this is literally asking people, do you consider yourself a morning person or an evening person? And yeah, that's a good example of the trade-off with really detailed activity or polysomnography data that would be nice to have, but you can't really do that for a million people. I've forgotten the sample size actually for this one, but say for the insomnia GWAS, I think this was 2.3 million people in this GWAS, which gave rise to 500 risk loci, which actually I think in a way for this schizophrenia counts isn't actually that many considering the sample size. So it's actually insomnia is actually estimated to be one of the most polygenic traits after depression and educational attainment. So the chances are to really say that we fully understood the genetics, that we can part of all of the common genetic variants that contribute to insomnia, we probably need samples of close, over 10 million. But when you look at the genes that come up and the pathways that these genes map to, synaptic signaling and neuronal differentiation, the SNAP25 gene that we mentioned comes up in the insomnia GWAS. You see enrichment in striatal and hypothalamic neurons, and yeah, strong genetic correlation with a range of psychiatric traits. We go on to that.
But before you move on, can I ask you, in these GWAS, how is insomnia defined? What you mentioned the phenotype that where natural.
And it's actually there's a bit of variation, but in this GWAS it is we give around 23 me and Imogen, I think. And I think this is self-report mostly, but there have been other studies that use, manually planned home, that they were much larger environments, where it's actually a diagnostic code. So that would map, and then quite highly intensified. But I think this is largely self-report, but I can't remember if it's I have insomnia or a doctor has told me I have insomnia and that thing, but yeah. But it does correlate quite highly, with when you look at the recipe, like overnight,
There's one thing I find in legal practice is that there's still probably people that come and say, I'm sleeping very, very, I don't sleep. And they can say to me, I haven't been sleeping for 2 weeks, and I know that's really very difficult. Someone be able to turn up and sleep and be able to function and not having slept at all. And then you would ask for a spouse or member of the family. But the person is not sleeping this, if they're not sleeping. And I remember I actually referred, one of them was particularly young, so I was actually a bit concerned and referring to the sleeping keys. And they said, well, actually, he does sleep 7 hours a night. But he wouldn't tell you that he was not sleeping. He would say, I said, what do you do? He said, oh, I'm lying in bed. But well, how do you know that you don't sleep? I said, no, I know that I will help. And I said, what do you think? Or if you say I lie there and look at the ceiling and I know I'm not sleeping. And I always found this group of people very, very interesting, even when the rest of the psychosis has, significantly improved. So it's not a sort of delusional stage but they will tell you, I don't sleep.
Well, I think some of the other side was sort of illustrate this as well, that, I think it can be definitely seen as a limitation to have just to rely on someone for data. And I think often we sort of say that's a limitation, but in some ways it can be a strength in that we're capturing a different, a slightly different phenotype. And I know from our work on sleep duration, is this the next slide? Yeah. My work on sleep duration, where we looked at, if you look at the correlation between, accelerometer derived sleep duration, but the one in on top rate 0.4. So actually, at the phenotypic level, the correlations can be a little high, but at the genetic level, they're quite different traits. The genetic architecture of how many hours someone is actually asleep in bed, as compared by, activity data, is a different trait to how people perceive their sleep. And you can make those comparisons, this is just sleep duration, but if you make those, well, I hope to eventually make those comparisons across more, sort of objective measures and subjective measures of sleep. And I think it's quite interesting to know which of those, probably both, but to what extent are each of those going to be important to explain the relationship with psychedelic traits. Because is it that there's some sort of healing process that's happening when you're looking at sleep that is protective for psychedelic traits, or is it that your reception of how well you're sleeping is the most important? But yeah, I think it is interesting. It does at the genetic level not correlate that either. I think some of the DSM slides also show that as well. Other sort of interesting things you can see, one of us said this, a prototype is also, again, quite genetically distinct from sleep duration traits. The genetics of how long you sleep doesn't match very well to when you sleep. I should say as well, the sleep duration data is also the way that that's asked in our data is how much do you sleep in a 24-hour period, including naps. So you're definitely there can be a really big heterogeneity within that of what might mean good sleep. It's less good sleep, but some of those people are sleeping 8 hours a night, going to bed, just falling asleep easily, and other people will be sleeping quite restlessly and sleep about the day, which is possibly why you do see a positive genetic correlation with short sleep and long sleep, and so both long and short sleep must be correlated to insomnia, because people may have insomnia but still actually sleep a lot of hours in the day, but it's been building up.
You mentioned my favorite word, which is math.
Oh, yeah.
Any person possibly, I have to ask. So there's every now and then you have a paper saying, having a short sleep after lunch is very good for you, you know, you do pretty much everything in your life. And then later on, you have another paper saying, oh, you know, sleeping during the day, very important, very bad. Well, Robin and I went to China. And well, I come from a country where this time of the year now, but you can see I'm trying to stay away this great show. But we went to China and we had a morning of lots of activities, and then we had lunch with colleagues in the general hospital, so we have not specific sort of outlook. And we were taken to a room, and the colleague in Chinese made some gesture, was switch off the light, close the door, and I'm not sure what was supposed to be really dark or sitting in the room. And then next door there was the junior doctor or looking after the patient who was actually sleeping with their pillow. She would take out after lunch or a drawer, have a 20 minutes now, off you go and work. So the entire hospital, including guests, was supposed to take it out at the 20 minutes. So and also I was speaking to actually colleagues who are Chinese, and you know, with the and then say that from school, Harry, I don't know if you're home with the thing, but certainly in that time, so home we don't yeah, exactly. You're on the other end of the spectrum, but you know, they would say that the primary school children would stop at an hour throughout school, even if and so what do you think people really enough? I mean, you know that.
Short 20-minute nap, or it's probably and that's nice brain studies showing that, you know, there's been probably degrees of this, sort of default, you know, which is kind of relaxation and daydreaming. And then the executive, load network, and a short nap has been shown to switch the brain in default mode to executive, which is why people make more focus and able to function better after a week nap. So, and I think you raised a very interesting point. I'm not sure it's about really good. I mean, sleep is a dynamic, very flexible behavior. There is not a fixed sort of state. And occasionally nap is fine. Occasionally performance is fine, as long as you know what the consequences. So, you know, if you do more much as a student, you know, you're driving a car the next day, you need to know that this is making it not smart. You're impulsive, you're probably using original, not a good time to make certain decisions in advance. You know, so it's I don't have partially take this is why this is natural. We start getting it from being such bad decisions and so well, I think it does help with what we know, in the sense that you are not less reflective, you become more irritable. And what happened with the later years, and I spoke to one of the advisors and said, yeah, you've got to start so, and you don't know it's there. But the very interesting thing is that she died with severe dementia.
Yeah.
And none of the sort of the whole stuff to show you that the groundwork is packed up and clear whilst we sleep, and you don't sleep, you can see it because it was a big groundwork in the CSF. I wouldn't say that more sleep is the cause dementia, but if you're on that risk spectrum, then it could well be, and we can afford, and she got it put up to she from kindergarten. Well, that's very good. Now that's interesting. So,
So yeah, just carrying on with genetic correlation, if this figure sort of nicely summarizes the extent of high genetic correlation between sleep traits and different psychiatric traits, with red indicating positive genetic correlation and blue being negative correlation. So you can see insomnia has the sort of broadest psychiatric signal, with significant positive correlations with narcissism, anxiety, ADHD.
Well, that's the napping then.
Oh yeah, I was going to say that it's definitely a GWAS of napping. So,
So what does it correlate with these two?
well, positively correlates with depressive symptoms and probably ADHD. But again, this is where I think you have to think about the phenotype as well. Because again, you know, there's there's going to be a lot of heterogeneity in the type of people
But
that are
that's
living
what
within that.
might be with subjective well-being.
Oh, there you go. Yes, it's another example of like a sort of U-shaped relationship. Yeah, so positive correlation. The other thing to say is that genetic correlation is just a sort of an average across the whole genome of where there are common variants, to what extent are they in the same direction, basically. So you actually could have very high genetic correlation when actually there's quite a small number of causal variants overlapping between the traits. So this is an example of a sort of, I often think of genetic correlation, it is often one of the first post-GWAS analyses that we do. And I think it's kind of like a good starting point of where you can identify areas of interest that then be delved into in more detail. I haven't included it here, but we used one of our recent papers that available genetic correlation analysis, where you, and then instead of just looking across the whole genome, what extent are the variants operating in the same direction, you can look at sort of partition the genome into much smaller blocks and see then if there's any areas of where the effects are going in opposite directions and that sort of thing. And you can also use a method like Mixer, which I've shown here, which it then, instead of just saying how, to what extent are these common variants going in the same direction, it's actually like quantifying how much of the traits overlap in their genetics. And actually that shows when you look at sleep and psychiatric traits, that actually the genetic correlation analysis almost underestimate the extent of that genetic overlap. So up to 80% of sleep trait variants are shared with psychiatric disorders. And then we move on to the genomic approach for the episode. Yeah, so the final sort of major area of value that I think can bring is taking a small variant approach. So I think, we all know that the diagnostic boundaries between traits, psychiatric traits can vary and move, especially in the case of psychiatry, where the comorbidities is more removed in the section. Being able to kind of model traits together is really valuable. So there's a newer data take the newer methods of genomic stratification modeling, where you can actually take multiple traits, so data from GWAS on multiple traits, and model them together, try and see actually the structure of that genetic overlap. So it's kind of like factory analysis at genetic levels, and we worked with just some statistics, and we can then pull out some latent factors which might themselves be a bit more informative in terms of the biology, especially when you're thinking about how that relates to psychiatric traits. So here's not probably focus the one, but genomic segmentation for sleep traits, took 12 different measures of sleep traits and found that the best fitting model pulled out sort of 6 different latent factors which cover circadian preference, efficiency, alertness, sleep duration, insomnia, and sleep regularity. So these are sort of genetically separable measures. You can see how this is the combination of objective and subjective measures, and actually, if you look, they are not necessarily loading onto the same factors. So even when they're in some ways capturing the same concept of sleep, like when you wake up or how long you sleep, they don't necessarily genetically look that similar. What you can sometimes, when you do these, you end up with this sort of overall like P factor or something that doesn't seem to be a sort of like unifying sleep genetics factor, which I think kind of varies what you see even in genetic correlation analysis, where there's almost no genetic correlation between some of these sleep traits, which isn't really what you see in psychiatry, where it's high genetic correlation between all of them. a well-known, you know, there is a lot more genetic correlation. So what you can, in theory, do, and this is quite an emerging field, there hasn't been a huge amount of work done in this, with sleep, but you can then, once you sort of establish this latent structure, you can then say, well, how do these latent factors actually relate to southern biology? And that might help us understand a little bit more about the sort of, rather than just looking at these pairwise traits, which are almost limited by the way we define them. This is a sort of a bit more of a data-driven approach to group biologically group traits. So yeah, they showed 12 sleep traits with 6 genetically acceptable factors. And yes, they showed, yeah, this is what we said, but the yeah, coming back to what you were saying about the objective and subjective measures of sleep not necessarily aligning, that definitely is supported in the genetic data. We applied this same sort of idea to genomic SEM, but focused to a specific set of traits that we were interested in based on some of our other work. And you can see here that this is kind of just another example of where the way that you define the phenotype matters. We get sort of different factor structures, depending on how we define sleep duration, whether we considered it as just number of hours slept or if we're actually thinking about it.
You remind us how do they define, like, what is short and what is considered long sleep?
So it's like a binary variable of people who slept 5 hours or less, compared to a normal range sleep of 7 or 8 hours.
And this is, you know, sort of 18 to 6, you know.
Yeah. That's actually that's a very important point, that almost all of the GWs that we have is from adults. A lot of the daily sorts of cohorts, where, for example, the background is people aged 40 to 70. The sleep duration data that we use the biobank and the mini veteran program, the average age is actually a bit older than more veteran programs, so over 90% male. So there's a lot more to learn definitely about the genetics of these traits, more diverse in the cohorts. Yeah. We did see no set, yes, sir.
There are people that, I mean, we need to show lost in the moon.
That's not, it's kind of, it's capturing anyone who reports short sleep duration. So actually there'll be some people who definitely do feel that that's a sufficient amount of sleep for them, and other people that don't. We sort of didn't end up making huge progress with this, but we did want to try and see, because the question is asked, how much do you typically sleep in the 24-hour period? But we actually wondered if kind of a lot of what you're talking about light perception, we wondered if when people were recruited to the study actually made a difference, and it did actually. We didn't quite figure out how to incorporate that into our genetic analysis, but I would like to do that. It took quite a lot of time to try and we sort of created a score based on people's month of recruitment and the assessment center where they were recruited by, their sort of latitude, longitude, location, estimating the hours of daylight that they would have been exposed to. It was a kind of small effect size, but we definitely did see across the US and the UK that when people were recruited made a difference. So again, that would be very interesting to delve into in greater detail to kind of see, I guess in theory, people actually, I don't know how that's probably would be really interesting to see. But is it completely random across a 12-month period? I bet I'm not sure in terms of recruitment. Then when people are recruited, I would imagine there probably are points of recruitment at certain times of year. So that could that would be interesting to see.
It's just the tight. If you take check, so everyone's on patient, so it's always the same time. And yet it could be a 4-hour difference in the sunrise, the sunrise, and what's the potential, which is.
No, that's so true, because I guess in the US there's 4 different time zones, so you know kind of but that would be so interesting to then compare to the Chinese people. It feels like people would be almost forced to not necessarily align with their natural sleep cycles.
Sharing with people across certainly Europe. And then the early rises, the east, and the late rises, and of course, it's beautifully.
Yeah.
And early sunrise,
Yeah.
and then later, so it's all on the same contract. Yeah.
That's
It would
really,
be
that's
fascinating.
really, yeah,
I should, and I like to learn this, no.
yeah.
Actually, so most of the GWAS will be, don't know maybe in the veterans course is more, are they reasonably homogeneous by ethnicity?
Yes, so
But I think
yeah,
that
yeah.
we know.
Yes, yeah, I mean, our situation we work, we did that on data as well. But the numbers are, and the MVP is much more diverse than the bank, but it's still, it's about 80%, right, for the European orchestra.
I think that, I guess, the genes involved in gene development, so regulation of preservable species. Would you think that they should be reasonably preserved across ethnically?
And it's very, very possibly so between movement and students, the battle. And indeed, one sort of effort, the individuals that have ancestry, they, in many cases, if they're living in this type of United States, they're living and there's an argument that everyone should be taken listening to.
We find, I mean, I find this in people under my head, that is pretty much 100%.
And the interesting, I think, particularly is there are some interesting links between the
From the actual German route,
yeah.
extensively.
So-
And from point at some, the time when we hold was to give vitamin D to everybody in the patients needs, with particularly now, because we do that for cycle, we go through.
Yeah, that's it.
Yeah. Yeah, I mean, it's not much difference to me, but it was way more than a public. I don't know how to see the geography.
Yeah, in our master creation analysis, we showed the genetic correlation between, when we do a section spread analysis, it's almost one, so we don't really see that showing up yet, but, and again, I guess the same sort of logic, these are really highly conserved. But I think in terms of the genetics, there might not be that much different, but then in terms of the sort of genetic regulation of biology or even environmental factors, obviously, then there is going to be a difference. And actually, most of the existing GWAS literature hasn't done sex stratified analyses. We did do it, but partly because one of our cohorts was 90% males, so we were quite interested in sort of, well, firstly, we were interested that the two cohorts, MVP and environment, they were themselves very similar, actually. And so then we went into it from there, but actually that's definitely something for probing. And most US also don't include sex chromosomes. So again, we're missing, I think the X chromosome is 9% for genome or something like that. So we're actually missing a huge amount there, but there's various reasons why they're not included, I think just in terms of historical complexities of analysing them. But that's definitely an area of, future direction. Okay, so polygenic was the next thing. So PRS or PGS, which is a way of taking this GWAS data and actually applying it to an individual risk prediction. Again, I think we've touched on them a little bit, but polygenic score is a weighted sum of risk alleles across genomes. So it's aggregating the effects of these thousands of common variants into a single number that affects an individual's genetic liability. So the higher your PRS, the higher your genetic risk for that trait. There's multiple different ways of doing this weighting. I mean, the weights are based on the GWAS that we talked about. The different methods which have various pros and cons depending on the question and the cohort. But they can allow you to leverage these really large discovery samples and apply them to potentially a much smaller or even an individual level. And they can also be useful in research at least, as a sort of proxy measure of a trait that maybe you can't measure, but if you've got genetic data, you can get a sense of someone's genetic risk for heavy cannabis use or something like that, and one can sort of try and use that as a proxy measure. So they've got a lot of value in research. Several studies have been used to use polygenic scores now to probe this relationship between sleep and psychiatric traits in both directions. So again, I've sort of picked a few of interest. So one study from a group at UCL used polygenic scores for short sleep to predict depression in adults, and they found that short sleep was predictive of depression, but not vice versa. The depression polygenic score didn't predict a sleep.
You can use them all in the same way.
Well, this is two sample MR. Sorry, no, this is polygenic PRS, and in some ways, I don't think this would meet the criteria for we can use polygenic scores in a sort of single sample MR framework. But this was much more like just a prediction model. Luckily, someone else has done that patient so another study showed that insomnia was associated with worse self-reported well-being and sleep problems in patients with psychiatric disorders. So this was a sample of 8,000 patients, inpatients, psychosis in patients. So they were sort of saying that higher insomnia PRS did associate with worse outcomes in a group that's already at sort of clinical risk. And then again, a little bit of touching on how sleep can be maybe a sort of early predictor later disease. When you look in children and look at a high polygenic score can predict later onset of psychiatric disease, but in a sort of disorder specific way. So the ADHD and NDD polygenic scores predict difficulty in initiating or maintaining sleep and excessive daytime sleepiness, which I think makes sense. Whereas the anxiety polygenic score was more predictive of disorders of arousal and nightmares. So interesting. So there are different psychiatric traits associated in different ways with the sleep traits. And then this study sort of looked at chronotype and insomnia polygenic scores, and then saw that those were predictive of schizophrenia, bipolar, ADHD, and MDD. And then they looked at a few others like eating disorders and OCD, which they didn't see any association with. And then thought I'd highlight this paper in particular, partly because it uses data from that paper, but also because I think that there's been quite a lot of interest over the conference in sex differences and why sex specific genetic analysis would matter. So this is a group at UCL that looked at where the genetic liability for sleep traits could improve the predictive of outcome of postpartum psychosis. So this is a sort of quite a small study, and it's actually in patients with existing bipolar disorder. But they did show that higher genetic liability to be a short sleeper was protective of postpartum psychosis. This definitely needs replication. It was only published earlier this year, but I think it's quite an interesting example of where a polygenic score analysis can maybe help get at a mechanism. So there's sort of the speculative interpretation would be that if you're sort of predisposed to being sort of naturally a short sleeper, maybe you're less impacted by the sleep disturbance that comes from when you have a baby. And I think I don't think that postpartum psychosis is necessarily that well understood. Actually, there's a lot of different factors with, hormonal factors as well. But I definitely think that the sort of onset of interrupted sleep is could well be somewhere on the causal pathway.
My question is this all information bipolar?
Yes. This was so whether this is seen in the wider cohort will be really interesting. I'm not aware of that much data from postpartum psychosis. So that's I think that will be really interesting to follow up.
I think and we was there and so. What in general, it's just, this thing here. And that the city did counterintuitive as something that was this thing simply says as question.
Yeah. Yeah, I only know from the genetic level that we hadn't seen that much, but I wonder if this is probably an example where we're a bit limited by some of the cohorts that we have, where there's, I mean, a lot of the data that this is actually built on would be in, for example, women quite a long way past having children and postmenopause, also maybe even past the sort of area Greek time of great fluctuation. So yeah, I think
So that makes sense. It's around like 50 men would say it starts to do.
exactly, and actually the average age of this cohort is over 55 alignment. So so I think that probably is could be why it's not showing up. And again, yeah, we're doing some work with some younger cohorts now. The kind of little project on Steve with the Owlsback cohort. There's not well, I think the sample probably with the biggest potential to replicate some of these is going to be our future partner, which has got a much younger age and should have forgotten what that aim is, but over 1 million, I think they're aiming to recruit for that. But specifically that's from age 18. So in terms of UK replication anyway, I think there'll be a lot of scope to replicate some of the UK Biobank funding for younger ages.
Only just one slide.
Oh, that's right.
No, it's not your fault, it's just we've been chatting and interacting, but I think
Okay. Well, anyway, also adding the bipolar progeny score did actually improve clinical prediction, which again, someone asked on the first day and about whether we should be doing PRS for patients. I think I sort of said no, but I guess I'd be more nuanced that there is a lot of issues with translating polygenic scores to an individual. It's not obvious how you can just take a population-based metric and apply that to someone's individuals with absolute risk. It's been to work from our team to try and our department, Ollie Payne and Catherine Lewis, to try to work on some of that. Importantly, they basically don't work at all in non-European ancestry, so this is a major, scientific and statistical limitation, but also a health equity issue. So. What I would say is the issues that we have with polygenic scores are definitely not insurmountable, and I expect them to be, resolved in some time. So I do think that at some point they would have some clinical utility, particularly when added to other clinical measures. And then I have a whole fun section on the daily randomization that we'll have to save for the walk. I'll tell you about. I can't imagine what anyone else would rather talk to me about over drinks. But basically, there's some causal, there's good evidence for relatively. I think
why we prepare for repeat exposure. One question, Professor Walker, I want to ask you mentioned some of the conditions of commonplace environment temperature. And of course, we now need, but most of us live in an environment where you can set the temperature, which is going to be favorable, or you can put your parcels or with an asphalt. But I'm thinking about parts of the world where we still understudy the savings from agriculture. Places where people just put that way the light and they eat whatever they eat and they.
There were very clever architectural solutions. So for example, as in working painting, about to flex.
People Sicilian.
Yeah, and
Marble.
and it's interesting. One of the global warming. So one of the responses of the UK is to make windows.
Yeah.
Rather than thinking about intelligence.